Hi, thank you for the invitation. Very happy to share with you the story of Asterias. I always like to start with just a little anecdote that will help you remember our name and our logo. Asterias is a regenerative medicine company, and as such, we actually draw our name and our logo from the common starfish, Asterias rubens. We are a publicly traded company. I will be making forward-looking statements today, so I'll refer you to our SEC filings for the statements of risk in there. And I'm also going to share with you a couple of patient stories and just want to highlight that these are example stories and that not necessarily all future experience will follow the lines of these particular stories. So Asterias' 3 programs are really based on 2 primary technology platforms. We have our pluripotent stem cell platform, in which we're using the H1 embryonic stem cell line, which is one of the first embryonic stem cell lines ever derived, one of the best characterized lines out there, and meets all of the various ethical standards that have been put forward by various government bodies and scientific entities as well. These cells have 2 primary advantages. First of all, they're pluripotent, so that means we can make any cell type that we desire from them for the indication that we choose. And so in the case of our OPC1 program, which I'll spend most of my time on today, we're making a neural cell type for the treatment first of spinal cord injury and ultimately of additional neurodegenerative diseases as well. In the case of our VAC2 program, we make an immune cell type known as dendritic cells from that same pluripotent stem cell platform. The second advantage of pluripotent stem cells is that they're immortal, and so we can make these cells scalably, consistently from the same starting material, and use a single cell line for the entire life cycle of the product and have the product be available off the shelf. In our cancer immunotherapy platform, we're developing 2 programs that are both based on the same 3 fundamental technologies. So the use of the dendritic cells to target immune responses. We're using telomerase antigen, which is expressed in nearly every cancer and plays a really fundamental role in the ability of the cancer cell to divide. And then finally, we're using a signal sequence known as LAMP to optimize the immune response to allow for response by both CD4 and CD8 T cells. We've tested that in an autologous form in our VAC1 program, and we're now moving into a clinical trial in the United Kingdom of our VAC2 program, in which we make the dendritic cells from the allogeneic pluripotent stem cell source. So these are our 3 programs. We've been very fortunate to have good partners providing non-dilutive funding for our programs. So I'd like to mention that the trial results in the spinal cord injury trial that I'm going to talk about today were under a grant from CIRM for $14.3 million that provided matching funding for that study. And we've also been very fortunate for our VAC2 program to have a pretty unique partnership with Cancer Research UK, where they are actually the sponsor of our trial that's initiating in the United Kingdom and are bearing the full cost of both the manufacturing and the trial for that as well. For our OPC1 program, we're in a phase 1/2A, and I'll give you a pretty detailed update on that today. I'm going to spend just another moment on this slide on our immunology programs because in the interest of time, and because we had several exciting announcements with respect to OPC1, I'm not going to talk further after this slide about those programs. But in VAC1, we have a phase 2 trial where we've looked 4.5 years out and seen in the maintenance setting in AML long-term relapse-free survival in 58% of patients, which is well more than expected based on many other large controlled trials in similar populations. And then in VAC2, as I've mentioned, we announced a couple weeks ago we have all the regulatory clearances to move forward now with our first in-man trial, which will be in non-small cell lung cancer in the United Kingdom. So now moving on to a more detailed update on our spinal cord injury program. So AST-OPC1 is a product in which we take the H1 embryonic stem cell line and we make a neural cell type known as oligodendrocyte progenitor cells. These are a support cell in the central nervous system. Their primary role is to mature into oligodendrocytes and to secrete the protein known as myelin, which is responsible for creating the insulating sheath on axons that functions much like the rubber coating on an electrical wire. So it prevents those electrical signals from shorting out when conducted over long distances. We've seen multiple mechanisms of action for this product, including both remyelination but also reduction of cavitation, promotion of neurite outgrowth, stimulation of new vessels, and importantly, those translate into improved motor function both preclinically and, as I'll show you today, we think now clinically as well. Our lead indication is in spinal cord injury, which is a very significant unmet medical need with a very high cost of care and a lot of additional lost productivity as well from disability, where very few of these patients are able to go back to work even many years after their injuries. However, the mechanisms of action that we've seen for OPC1 we believe do have application in many other neurodegenerative conditions as well. So this is just a brief slide showing some of our very large body of preclinical data. We have seen remyelination in relevant models and rodents. We've seen that when we put OPC1 conditioned medium on nerve cells in culture, we do see outgrowth of neurites. And when we transplant OPC1 in rodent models of spinal cord injury, we see that the cells actually fill the area that would otherwise be an injury cavity and allow for those myelinated fibers to extend across the area of injury. And these results have been published in multiple publications shown at the bottom here. Our current study is called the SciStar study. We have a total of 5 cohorts in the study. The first 3 are in the most severe category of spinal cord injury. These are known as AISA injuries. And these are patients who have both no sensation and no motor function below the level of their injury. We've done 3 dose escalation cohorts in this population, starting with a safety dose of 2 million cells and then moving on to 2 doses designed to bracket the predicted efficacy range with 10 million and 20 million cells. Once we had some safety data in the AISA patients, we were actually able to expand the study by adding 2 additional cohorts of patients in slightly less severe injuries known as AISB. These are patients who have some sensation but still no motor function below the level of their injury. And in these patients, we're testing just the 2 efficacy doses of 10 million and 20 million cells. We've accomplished quite a lot over the past year. We actually, in July and August, completed enrollment and dosing of cohorts 3 and 4. And we've now also dosed 3 patients in cohort 5. So we're getting very close to full enrollment of the study. As an indication of the very strong safety data that we've seen so far, we also recently received FDA's permission to expand our inclusion criteria to include patients with injuries at the C4 level of the cord. And that's a population that we were originally precluded from going into due to potential safety concerns. Should the injury be ascending through the cord, those patients could end up on a respirator. Because our safety profile has been good so far, we have now been able to extend into those patients as well. And we've recently opened an additional 3 sites, bringing us to a total of 9 sites now enrolling in the trial. And we just announced on Monday that we've received our MAT designation as a result of the data that I'll share with you today. So before I go into the data, I just want to orient you a little bit on the measurements that we're using in the trial. So we're looking primarily at upper extremity motor function, so ability to move the muscles in the arms, hands, and fingers. And we're doing that using 2 measures from the same international exam known as the INSCI exam. That's the major exam used to classify all spinal cord injury patients. The muscle groups in the arms, hands, and fingers are classified on the INSCI with what's known as the upper extremity motor score, where each muscle group is rated from 0 to 5 for a total score of up to 50. And then those are used to calculate motor levels, which are considered more clinically significant because these are the measurements of the level within the cord at which you have clinically useful function and above which you have normal function. And so we have defined as the endpoint that we're looking at most closely the improvement of motor levels on the INSCI exam. And we've done that based on results that were published by John Steeves, where he showed that these have very direct translation into clinically meaningful benefits in patients' ability to care for themselves. So, for example, on this slide, a subject with a C5 injury would not be able to perform any of the activities of daily living shown here fully independently, and therefore would need 6 to 12 hours of daily assistance. If you can improve that patient by 2 motor levels, you can take them to a C7 level of injury, as shown in the right-hand column, where they have near total independence on all of these activities of daily living, and would only need about an hour of help daily with their housework. So you can imagine that this also translates very much into benefits in cost of care as well. There also are other benefits to regaining arm, hand, and finger function that aren't shown in the table in the Steeves paper, but we all know how important being able to type emails, use our phones, those sorts of things are in modern life, both for our day-to-day life and also for our ability to be productive members of society. And so a couple of the individuals in our trial who have regained motor levels of function have also seen benefits in things like their ability to write emails and use their phones as well. So when we first designed this study way back in 2013, we set as our target to have at 12 months 45 to 50% of our subjects improving 2 motor levels. And that was based on the work of John Steeves, where he showed that spontaneously you would expect 26% of this patient population to regain 2 motor levels, and that if you could improve on that by about 20%, you would have the ability to statistically power a trial with a very reasonable number of subjects. We've now also gone back and developed our own matched historical control set to make sure that the inclusion criteria in our trial were not in any way biased relative to Steeves's data, and we see a very similar result of 29% of the matched historical control group improving 2 motor levels. And we had reported previously that at 9 months after injury, we were hitting that target with 50% of our cohort 2 subjects improving 2 motor levels. So now, as of 12 months, we've actually further improved. We've got 67%, or 4 out of 6 of the patients in this cohort improving 2 motor levels. And importantly, we're also seeing improvements over the control group on a number of other measurements as well. So this is just that in graphic form. So 67% of our patients exceeding both the 29% in our matched historical controls and 26% as published by Steeves. We also see a similar improvement over time in the upper extremity motor score as well, where we have an average of 12.3 points of gain as of 12 months as compared to 7.8. And this translates in every patient we're seeing at least 1 motor level of improvement on both sides of their body. We've looked for evidence of whether we're getting a stable tissue graft in the patients using MRI methods that were actually developed by our chief medical officer in his thesis work. And we do see in all of the subjects in cohort 2 the signal on T2-weighted MRI that is suggestive of durable engraftment at the injury site. And as I mentioned earlier, we just on Monday announced that we've gotten our RMAT designation, so we're very excited about the potential of this to allow us to work closely with FDA going forward on our clinical development path. So in summary, I didn't have time to talk about cohort one today, but we also saw very strong safety in that. And importantly, those patients actually tracked quite well with the historical controls. So we are seeing evidence of a dose-dependent effect between cohort 1 and 2. We do have a strong safety profile for OPC1 across actually a total of 29 patients in which it's been administered between the previous safety trial and this trial now. Importantly, this is something we don't think has ever been seen before in spinal cord injury. We're seeing prolonged effects out to a year after administration. So we're really looking forward to moving forward, collecting the rest of the data from the current trial, and designing our next trial as well. And I think I'm out of time, so I'll leave it there.